on? Okay. Good morning, everyone. For anyone who doesn't know me, I'm Mark Kwiatkowski, and it's my pleasure on this last day of the Living Machines conference to introduce Andrew Schwartz, who comes to us from Pittsburgh, Pennsylvania, which I know is a wonderful city, having been born and raised there. But before arriving in Pittsburgh, he got his PhD at the University of Minnesota, and then did postdoctoral work at Johns Hopkins University and at the Barrow Neurological Institute in Phoenix and the Neurosciences Institute in San Diego. And over all these years, he's been developing really an amazingly, increasingly sophisticated series of cortical bionic interfaces with steadily increasing numbers of degrees of freedom and, well, and grace. And so it will not surprise you that in addition to his many publications and awards, he's also captured the popular attention, and he's been appeared everywhere in BBC and New York Times and everything else. And so I think this is really the perfect plenary talk to kick off this last day of the Living Machines conference, and I very much look forward to giving him the floor and letting him tell us what he's up to.
Thanks, Mark. I also want to thank the organizers, Tony and Anna and Sylvia and Paul for this meeting. It's a great meeting. It's in a really spectacular venue, and the city's been incredible. The dinners have been wonderful. Everything's great, so thanks a lot. So I'm going to give a little survey of what we've done over the last 30 years and how that outcome has led to practical application. But I want to try to emphasize a bit of what's under the hood to give you both an intuitive feel and a little bit of knowledge and a little bit of the way I think about this as a scientific problem. So if we just start from basics and we ask, what happens in the brain when we generate a movement? The classic idea is that the brain is composed of individual components, much like a machine, with each component operating, doing its own little thing semi-independently and sending its output to the next stage. But if you really look at a brain that's operating in a real environment, fully intact, awake and behaving, which, by the way, is very, very rare in the field of neuroscience. 99% plus of neuroscience is done in vitro, okay, or in reduced preparations. So the number of experiments that are done in extremely realistic settings is very, very few. So anyway, let's say that we had a magic machine and we can look at individual neurons firing in the brain during a volitional task. And in this case, the volitional task is we ask a subject to draw with their hand. Just draw a circle like this. And we say, what would the brain activity look like during this type of behavior? Well, it would look something like this. You would see massive activity throughout the entire brain. And when you try to look at the pattern of activity, it would not be very obvious. Okay? So every time we generate a behavior, there are billions of neurons that fire more or less simultaneously within the brain. And we don't really have a very good understanding of any of the principles that govern that activity. And just to take one step further down, the element that we're really interested in here is the neuron. The neuron is sort of what we think of as the building block of neural processing. And neurons have cell bodies. They have inputs from other neurons. Right here, these are synaptic inputs. These synaptic inputs are integrated, and when enough of them occur simultaneously, an action potential is generated, and that then this pulse is transmitted to other neurons. And the way we're going to listen to these neurons is by placing an electrode in the extracellular field that's generated when an action potential occurs. So we put a microelectrode in the vicinity of the neuron to listen to the electric field that's generated when action potentials are fired. And so the experiment that I want to start with is exactly what I showed you with that puppet. This time we have a monkey that's trained to draw with his hand in space like this. The monkey's looking at a virtual reality image of what his hand's doing. It's three-dimensional. He puts his hand in this virtual object and just traces it like this. And as he traces it, we listen to neurons fire in the monkey's motor cortex and just make the attempt to see if there's any kind of principle that we can ascertain from the way these neurons fire. And it turns out it's very obvious. One of the aspects is very obvious. So I'm going to show you this early experiment. You see an action potential firing right here. Here's the monkey's arm moving. And you'll notice there's a background activity when he's steady, and it's highly modulated as he moves his hand. And in this case, the neuron fires when he moves upwards. The neuron increases its discharge rate, and when he goes downwards, it shuts off. Very clear modulation. And so what appears to be driving this neuron is the direction that the arm happens to be moving. And this is one elaboration beyond the initial finding that was generated by Apostolos Georgopoulos at Johns Hopkins in the early 80s. And what he showed is that if you have a monkey operating a draftsman arm on a tabletop, and inside the tabletop are a set of lights, so that the monkey starts at the middle and moves to each of these lights individually when they're lit. So this is start at the center, move to one of these peripheral targets. This is what's become known as the center-out task. And the parameter that's being tested in this case is movement direction. So during each movement, the direction is fairly straight, it's constant, and all other parameters are much the same. So the variable in this case is movement direction. And if you plot that on an axis down here, and you record a single neuron, much like I showed you before, so this is firing rate, you'll find that there's a single direction right here that the neuron fires the fastest in. And as these are the data points, the firing rate of the neuron as you move in these different directions, you'll see that you can fit this function, this tuning function, with a cosine function. So we call this the preferred direction. It's the direction in which the cell fires the fastest. And as you move at angles away from that preferred direction, the discharge rate changes systematically. Now, this relationship has been found throughout many neurons, not just in motor cortex. Most of the neurons in motor cortex have this relationship. But as the years have gone on, in the last 40 years now, this has been found throughout the entire neural axis, from the periphery, okay, to visual systems, to subcortical areas, cerebellum, basal ganglia, superior colliculus, different, AIT, face cells. They all have this general cosine relationship. So it's pretty robust throughout the nervous system. And I just want to take a little bit of time to delve under the hood about what a tuning function like this might mean, or at least the way I think about it. So we can call this an encoding function, because on the left here we have firing rate. On the right we have these variables that we think influence the firing rate. Okay, so this is a very simple relationship. What I just showed you was a two-dimensional task, and we just have this simple planar relationship. So we have movement direction, and then we have the slope in each axis of the plane, which we can fit with a regression coefficient. So you can't have a simpler model of firing rate, okay? And we can rewrite this equation in terms of vectors. So we have one vector here, this B vector, and we have another vector, this M vector. The B vector is the preferred direction of the cell. That vector points in the direction that the cell will have its maximum firing rate. The M vector is just the variables, the direction that you happen to be moving. And so what we have then, if you rewrite this equation, is just a dot product between these two vectors. Okay. Exactly the same equation. We can rewrite it. Dot product has this form. And here we have this cosine of theta, which is exactly what I showed you in the plot in the previous slide, okay? So it basically makes sense. And the way I think about it in my mind is I think about a unit vector in the movement direction, a B vector in the cell's preferred direction, in this case, straight up. The cell will have a preferred direction straight up. I project this vector onto the B vector, and the length of the B vector is the firing rate of the cell. And as I move at angles away from that, okay, you can see that the projection, this firing rate now becomes very small, which means that the neuron's firing at its background rate. And if I go opposite the preferred direction, the neuron actually begins to shut off, okay? But the important factor here is that the only thing that we need to worry about is the angle between these two vectors. And this is actually the geometric equivalent of correlation. And you might ask, or at least the way I think about it, what does the cosine function mean? It means that these neurons care about how well the input to these neurons matches what this neuron will tend to do. In other words, you say, how close am I to the maximum firing rate of this cell? And this is just the angle between them, or how well correlated is this to the preferred activity of the cell, the preferred set of parameters that makes the cell fire. Now, the handy thing about this is that I'm going to show you that I can continually increase the dimensionality of this movement vector. So instead of, first thing I'll do is go to 3-dimensional space, and I'll eventually proceed all the way to 10-dimensional space. But the important thing is that the form of this equation stays the same. So it's a very simple linear equation, even though I increase the dimensionality of the description that I'm trying to make here. Okay, so I just showed you the encoding function. This tells you what makes a neuron fire. For neuroprosthetics, we're really interested in the inverse. We want to look at neural activity and decode it to try to understand what the subject's intention is. And the way we do this is with a population vector. So each of those preferred directions I showed you before is represented by a blue vector. I record from many cells at the same time, each of which has a different preferred direction represented by these blue vectors. And the length of the vector is how fast the cell is firing at a particular point in time. So you'll see this is a snapshot of a movement, and some cells have very long vectors, some cells have very short vectors, but they're organized so that if I take the vector sum of this, the resultant vector is this yellow vector, it's the population vector, and that's my decode. Okay, so what I'm saying is that at this particular point in time, I decode the neural signal, and the intended movement direction is this yellow vector. This is what we call the population vector. I can do this continuously throughout a movement, and when I do this, these are real data from real monkeys that were drawing with their fingers, and I can actually get a high fidelity representation of the movement. There it is right there. And you can clearly see that the monkey was drawing a spiral from inside out and outside in. So I get this high fidelity isomorphic representation based on this very simplistic encoding and decoding model. Okay? So that's really what's under the hood. This is the way I decode my neural signals. Now, the take-home message from this is that I'm not just simply getting movement direction, but I'm actually getting movement velocity. So I'm getting both the direction and the speed from this reconstruction. And when I hook these vectors up tip to tail in this integration process, I can actually get position, or in this case, a representation of the trajectory. Now, it turns out, for those of us that study movements, that trajectories are extremely important. And the reason is, again, think of what a trajectory is. It's a time sequence of points in space, okay? And the way I like to think about this is trajectory is behavioral output. So the only way you know about what's going on inside my brain is by the way I express my thoughts, and that's through movement. Whether I'm gesturing or speaking or pacing, okay, what I'm doing is generating trajectories. And I just want to spend a little bit of time talking about that. So when we move, there's characteristics of movement, these psychophysical invariants. And laws, there's actually rules that govern the way we move. And these were initially described for handwriting. In this case, I'm going to show you some drawing experiments. And it turns out that when you move, when you draw, or when you generate trajectory, there's a relationship between the speed that you move and the curvature of the trajectory. So what happens is that when you're moving straight, you move rapidly. When you come around a corner, you slow down. And so if I draw this figure 8 here with my finger, here's the trajectory. I slow down for the curve, and I speed up on the straightaways, and I slow down. So you can actually break these continuous trajectories into segments based on their movement speed. In this case, this is what the monkey drew. This is what I decoded from its brain. Okay, so I can get this neural representation of the trajectory. Now, if I break these into segments, so I had the monkey draw 8 different lemniscates, okay, different orientations, and I decoded his brain activity, and this is what I got. And the colors match the segments here, okay? So I can get this, not only can I get this isomorphic representation, but I can capture the dynamics of the movement very well. Now, it turns out that this segmentation law is actually even more detailed. So that if I plot velocity against curvature, I get an explicit law. And this is actually the speed of curvature is related to the 2/3 power of curvature. It's a power law. And this was, as I said, initially found in handwriting. But as years have gone on, it's been studied for all movements. And it's actually so strongly represented that it's really called the law of animate movement. If we look at some object that moves, if it obeys a 2/3 power law, we consider it to be an animate movement. If it doesn't, we think it's inanimate or machine. So, you know, if I want to demonstrate a robot, it might move like this, right? And the reason that you would think that's a robot is because I'm not obeying the 2/3 power law when I do that. If you do an experiment where you have a blinking light in a dark room at the fingertip of someone that's drawing something, and that light's going at a constant velocity, you can clearly see the shape that the subject's drawing by following the blinking lights. But if I play a trick so that I change the frequency of the light on either side of the circle that I'm drawing, so let's say I speed up over here and over here, okay, and I'm still drawing a circle, but now the frequency changes over here and over here, you'll perceive that as an oval instead of a circle. And the reason you do that is because you think it obeys a 2/3 power law. It appears that you slow down for the curves. You know from your life experience that when you go around a curve that you slow down. So you perceive that as an increase in curvature. If you study movements of children, when they first begin to move, don't obey the 2/3 power law. They have a power law, but the exponent is noisy. But by the age of 12, they settle down on the 2/3 power law. Okay, the exponent becomes 2/3. So this is a very, again, another robust example of a law of movement. And it turns out we can also capture that in our neural signal. So if I look at if I plot curvature to the 2/3 power against angular velocity for those movements, both the actual finger trajectories and the neural trajectories, I get this linear relationship. Okay, so I can extract the 2/3 power law from my neural activity. So that means I can catch the natural features of movement from this extraction. One of the other things that's interesting, if you just look under the hood a little bit, is you look at eye movements during this tracking. And you would think that, you know, since the subject or the monkey has to stay within this template, and he has to move around 5 times before he gets rewarded at the end, that he'd be very careful about making sure his finger stays in this oval. So you would think that the eye movements would follow exactly what the trajectory does. Here's the eye movement, and this little blue dot here is the finger movement. And look what happens as he starts drawing this. So there's the finger movement. What you see is that he saccades to the high curvature regions. Okay, he doesn't follow this. He jumps and he precedes the finger. So he foveates these regions of high curvature and waits for the finger to get there and then jumps to the next region of high curvature. So these curved regions of the trajectory are extremely important. And behaviorally, the reason for this is that he never makes a mistake when he's on the straight part, okay? He always makes the mistakes. He always falls out in the curved part. So he learns that he needs to pay attention to those curved parts. If you look at somebody in a driving simulator going down a road, and they saccade to the curved regions of the road. They pay particular attention to the curved regions of the road. Now you think, how does this make sense in control systems? Well, imagine that your brain is a direction processor, and you have to transmit these bits of information through a fixed channel to execute the movement. If you're in a straightaway in your directional processor, the information is just keep doing what I did in the last time step. Okay, if you're going straight, that's all you need to say. However, if you go around a curve and you have a high and you're changing direction quite rapidly, you need to specify that direction very precisely. And so it requires more bits of information. If you have fixed channel capacity, you have to slow down and wait for those bits to be transmitted. And so this all makes sense in terms of this 2/3 power law. So you can gain some insight into looking at the behavioral consequences of these tasks. Again, if you look at this and you look at hand position, the density plot, you see that the hand spends a lot of time in the curved regions of the trajectory, and you see that the eyes also spend all their time in these curved regions. Okay, so what I'm trying to emphasize again is the behavioral impact of these movements. And again, going back to this idea that trajectories is really behavioral output, I like this passage from Moby Dick when he's talking about, when Melville's talking about Ahab. And of course, Ahab is completely infatuated with Moby Dick, the great white whale. And so there's this passage where Melville's talking about Ahab on the deck of his ship. And he says, "You'd physically gaze upon the ribbed and dented brow. You would see strange footprints, the footprints of his one unsleeping, ever-pacing thought. That thought would turn in him as he turned, and pace in him as he paced, completely possessing him, that it seemed the inward mold of every outer movement." Okay, I think that expresses much better than I can what I'm trying to say here. All right, so imagine that we want to do neuroprosthetics. We want to capture this intended movement and have all the ingredients of thought, as much as we can, extract within that movement. How would we do it? Well, we need to record from a population of neurons simultaneously so we can perform our decoding operation. And the way we started this many years ago was with these microwire arrays. Very crude, 50-micron wires. We cut them with a pair of scissors. We made them ourselves and allowed us to record multiple neurons simultaneously. And the task that we used again was this virtual reality task where we had a monkey looking in this 3D monitor, and he would see this target array. He would start from the middle and reach to the corners of the cube. So this is now a center-out task, but it's in 3 dimensions, okay? And so he would see this target floating in front of him. We'd track his hand. That was a cursor, and then he had to move his hand to each of these targets in virtual space. And what we did is we started out by having him move his hand to these, and then in the middle of the task, we would switch the cursor off and decode in real time this intended movement signal and move the cursor with that. So this again now is, circa 1997, and I'll show you what it looked like. So here it is, the monkey. Here's the cursor on his hand. Here's the targets, and you'll see initially he's moving to these targets that we superimpose the screen display on this display. And then we switch, these big targets are now brain control. He keeps moving his hand to those, except I think this one right here, you see he didn't move and the cursor still went there. Eventually he just drops his hand and just moves the cursor just by thinking about it. Okay? So these were early experiments done by Don Taylor at Arizona State University. And I just want to show you how we trained. So initially, we didn't know what the cell's preferred directions were, okay? And we basically had to do an iterative process where we kept showing the monkey the target. We would assume that was our movement direction, and then we tried to regress, get these regressors from these movements. And you can see it's an iterative process. It took about 15 minutes. I'll show you some snapshots. So this is what it looks like when we don't know the cell's preferred direction. Okay, the cursor doesn't go anywhere near the target. So we do a few of those. We re-estimate the preferred direction, and we do a little bit better. So this is, now he's at least getting toward the target. And we, again, do this iteratively. And if you wait, we go to the next step. It gradually gets better. And at the end of 15 minutes, after several iterations, the monkey can actually get to these targets quite well. So now you can see he goes straight to the target. He can go back and forth, and he's very efficient at moving to these targets. And this was, again, 15, 20 years ago. Now in the lab, you come to these monkeys can just snap to these targets very rapidly and almost perfectly. Okay, so let's just think about what's going on here. From an engineering point of view, we like black boxes. So think of the brain as a black box. What the job of the brain is to generate behavior. This is intended behavior. We capture that with our electrodes and in the form of a population of neurons. We apply our extraction algorithm to get, in this case, a population vector. This is our estimate of intention. We feed that to a prosthetic device. I'll show you in a second. This will be an arm or a hand. And what I just showed you was the cursor. And that moves and provides feedback to the animal. The animal can actually see whether it was correct or not. And of course, since this is a model, it's always going to be wrong, okay? But because this is going back to the brain, the animal has a chance to correct it. So what he can do is change the way his neurons fire to make this work better, okay? This is learning. This is learning. We can actually study it very well right here, okay? And that's a different topic. But the point is that by changing the neural activity, the animal can increase its performance, or the subject can increase their performance. And this is extremely important. This is the whole reason that these prosthetics work. It's because we let learning take place. And what precisely do I mean by that? Before I get into that, I'll just show you that this works. So if you look at performance or success rate, how small can the target be and the animal still gets there? You can see that over 40 days, his performance continuously gets better. Okay, so he's actually learning. His success rate goes up. If you look at what's going on here, in this case, it's a three-dimensional tuning function. And what we're trying to do is regress. We're trying to make an equation that's accurate. And the accuracy is going to be guaranteed by how well we fit our regressors. And, okay, in a multiple regression, we can quantify that by R squared, variance explained. And what I'm going to show you is that if I plot variance explained at the same time scale over here, so now this is model fit, or R squared, it exactly matches the performance, okay? So what this means is that as the animal learns the model, okay, learns to make his neurons fit our model, his performance goes up, okay? And this is very clear. Everybody that does prosthetics in this way has found the same thing, and it's extremely important. Okay, so we have two things going on. Okay, we're trying to fit with our model what goes on in the native brain, okay? And at the same time, the subject is learning what our estimate is and trying to fit their activity to our model. So we fit our model to the brain activity fits to our model. Okay, it's a two-way street. All right, so over the years, we've improved our technology. So instead of using these crude microwires, we now use these silicon devices called Utah electrode arrays. This is a 4x4 millimeter platform. There's 100 electrodes that are carved out of a solid chunk of silicon. These are 1.5 millimeters long, and they have platinum iridium recording tips. You turn this upside down and inject it into the brain. So here's monkey motor cortex here. Here's the nose. This is the central sulcus. We have one array in this case in the hand and arm area of motor cortex, the hand and wrist area. This is in the shoulder and elbow area here. So we have 200 electrodes placed simultaneously. And now what we're going to do is a different experiment. So this time we have these microelectrode arrays implanted in motor cortex. And again, just to review, what we do is we record each neuron individually. We estimate each neuron's preferred direction. We assemble these in a cluster to get a population vector. These are those blue vectors I showed you initially. We take a vector sum to get this population vector. That population vector is analogous to instantaneous movement velocity, and it's specifically velocity at the end of the effector, which in this case is an anthropomorphic robot arm. So this arm, what we're controlling in this case are 3 dimensions of movement, so the animal can move anywhere in its volume around him in the workspace. In addition, we added another degree of freedom that allows the animal to open and close the hand. It's just aperture, open and close. And so what we do behaviorally is we place food in front of the animal. He can reach out with this device, controlling it with his brain activity, grab a piece of food and bring it to his mouth. Okay, so now we're talking 2005, 2006. And specifically now, we've increased the dimensionality of our tuning equation by adding this 4th term, this aperture, so we're controlling 4 degrees of freedom. Now remember, the form of the equation is exactly the same. It's just 2 vectors. They happen to be 4-dimensional in this case, and the firing rate is just proportional to the cosine of the angle between them. So here it is. In this case, we're placing marshmallows out in front of the animal. And what you can see is that there's continuous control. So we're not turning this on and off. We're recording the brain activity continuously. So even while he's chewing, he's controlling the arm. And you can see he's very skilled at this. This is actually a pretty demanding task because there's no wrist and he has to be very precise when he grips this. Here's another example. We talk about embodiment, and I can't prove that it's embodied. I can just give you an impression. But in this case, he's so comfortable with this device that when he decides that he'd rather lick the marshmallow off the hand here, he just does it, right? So this isn't scripted. He's completely, volitional. And then when he decides he's had enough, he can just go and get the marshmallow. Okay. So here's another task where he's going for a piece of apple. So you'll see at the end, he didn't quite get it in his mouth, and he goes back and nudges it in. You can see that in the slow motion. Okay. So this was four-dimensional control. What we did next was add more dimensions, and we did that by adding a mobile wrist. So this is, again, a more or less anthropomorphic wrist. It has 3 degrees of freedom, just like your own wrist. And what we did is we placed target cylinders around the animal so that he'd have to reach out and orient the wrist to grab the handle. So we had these different targets out here for him to reach to. Now our tuning equation is increased. Okay, so we have our XYZ displacement, and now we have yaw, pitch, and roll of the wrist, and our aperture. So now we're controlling 7 degrees of freedom simultaneously with this signal. And here's how well the animal can do. Okay, so that works. Now, we're proceeding from proximal to distal. What about the hand? Well, the hand by itself has somewhere around 21 to 23 degrees of freedom just by itself. That's a huge control issue, right? Trying to control all those degrees of freedom. Now, it turns out for grasping behaviors, many of these degrees of freedom are correlated. Just imagine when you curl your finger, okay, you have 3 or 4 degrees of freedom. They all move at the same time. They're highly correlated. And so what we can do is, because of this correlation during grasping, we can do dimensionality reduction and describe grasping with a set of 3 or 4 principal components. And so the experiment that we did was to have animals use their own hands to reach for a variety of objects as we recorded neural activity. So we had these different target objects that we presented to the animal, buttons and handles and pins and cones and discs, so we could get a wide variety of hand shape while we recorded our neural activity. And what we did is we expressed these hand shapes as 4 principal components. Okay. They look like this. This was actually described originally by Marco Santello and John Sechting while they looked at human movements. Now, we got these principal components by observing monkeys. Monkeys don't move their thumbs very much, okay, so the principal components are slightly different. Now what we did is that we made our tuning function, okay, so we have our XYZ, our wrist, and now instead of just aperture, we have hand shape expressed by these 4 principal components. So now we're up to 10 degrees of freedom, okay. I don't know if any of you know other people that are doing brain-controlled interfaces, but the most that any other group has been able to obtain as a decode are 3 degrees of freedom, right? If they can get to 3 degrees of freedom, they're really excited. I'm showing you 10 degrees of freedom here, okay? So there's a clear difference between what we're doing and what everybody else is doing. Okay, and to show you that this works, what we did is we reconstructed the animal's hand movements during this task from neural activity. So we're actually showing you how well our model works. And here you can see the actual and the predicted as he reaches to each of these targets. And you can see that we're doing a pretty good job. Okay, so what I've shown you is that I validated all these models, right? I made these models, and then I've demonstrated that they work. Now, at this point, we'd say, well, we really want to make this something that humans can use, but we didn't really have, there were prosthetic arms out there, but there weren't really very good prosthetic hands. Recently, DARPA, the American Defense Agency spent about $130 million in this program. And one of the outcomes of this was this a modular prosthetic limb that has 17 degrees of active control, 26 degrees total. So again, you have 3 at the shoulder, you have the elbow, you have 3 at the wrist, and now you have something like 12 or 14 at the hand. So this is a pretty decent hand. Each of the fingers can curl, you have ab and adduction, you have the thumb, and the great thing about it is you can have thumb opposition, right? A uniquely human characteristic. The whole arm weighs 9 pounds. We talked about, hydraulic versus electric. Anyway, the objective was to make this 9 pounds with a removable battery. The thing can lift 50 pounds, okay? So this is a pretty sophisticated arm, and it's the size of an average adult male human arm. So here's what the hand looks like, all right? So now we have all the ingredients that I think we need. We have a validated model, we've developed the recording hardware, the decoding algorithm, and now we actually have a sophisticated effector. So at this point, we felt we were ready to try this in a human. So in December 2012, we implanted our first human. Her name is Jan, and she was 53 years old. She's been paralyzed for 11 years. She couldn't, at the beginning of the study, she couldn't move anything below her neck. Okay? And I'm just going to show you a composite of newsreels that sort of tell the story perhaps better than I can. So you can sit back and watch the movie.
It's taken many years of working with monkeys to develop the technology needed to record from neurons involved in arm movements.
The monkeys have brain control over this robotic arm to move it forward and grab a piece of fruit as it's presented and then bring it back to their mouth to feed themselves.
Incredible as it may seem, these monkeys learned to feed themselves with a robot arm that was being directly controlled by their brains, as if it was simply part of them.
This is a biofeedback, closed-loop kind of experiment, and that there's almost an automatic learning that's going on.
Schwartz showed us how he implanted tiny sensors like this one into the brains of monkeys and then wired them to a crude robotic arm. Schwartz told us that when the monkey thinks about moving his own arm, his brain cells or neurons fire off electrical signals. The sensor in his brain can pick up these signals and send them to the robot.
So he's operating the arm in 3 dimensions, up, down, forward, and back.
As well as the gripper.
Well, what you're telling me is that the monkey is operating this arm with nothing but his thoughts.
Absolutely.
What are the chances that a human being would be able to do the same thing?
Oh, we think a human being could do much better.
In a decade of war, more than 1,300 Americans have lost limbs on the battlefield. And that fact led the Department of Defense to start a crash program to help veterans and civilians by creating an artificial arm and hand that are amazingly human. But that's not the breakthrough. We don't use that word very often because it's overused. But when you see how they have connected this robotic limb to a human brain, you will understand why we made the exception.
One day I had trouble. In the evening, I was making a lot of trips in and out of the car. It felt like my legs were dragging behind me.
Within 2 years, a genetic disease called spinocerebellar degeneration broke the connection between brain and body. Now at age 53, Jan Sherman can move only the muscles in her face and a few in her neck. A neuroscientist just across town at the University of Pittsburgh was imagining how people like Jan might be restored. Andy Schwartz, on the right, is working on an ambitious Defense Department project called Revolutionizing Prosthetics.
What I'm going to do right now is I'm just making some marks here in your hair.
The procedure was done by University of Pittsburgh neurosurgeon Elizabeth Tyler Kabara, who showed us that the area that controls hand and arm movement is accessible right on the surface of the brain.
May I have some irrigation?
During the 6-hour surgery, 2 sensor arrays, each the size of a pea, were placed on the surface of Jan's brain.
Woohoo!
Then they were wired to 2 computer connections called pedestals, the gateways to Jan's thoughts. 5 months after the surgery, we came back to see whether she would be able to control the robotic arm with nothing but her thoughts. They plugged her brain into the computer, and this is what we saw.
I can move it up and straight down and left and right and diagonally. I can close it and open it, and I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over
Okay.
there. Let me grasp your hand there. There we go.
Oh my goodness. Wow.
And I can do a fist bump if you'd like.
That's amazing. What are you doing, Jan? What's going on in your mind as you're moving this arm around? What are you thinking?
Okay, the best way to explain it is raise your arm.
Uh-huh.
Go. Now,
Right.
what did you think about when you did that?
Well, not very much. I do it all the time.
Exactly. It's automatic.
Is that hard work? Are you having to concentrate?
No, it was hard work getting there. I struggled greatly to go up and down at the beginning. Now up and down is so easy, I don't even think about it. Side to side, don't even think about it.
Just like
Just like that.
your arms used to.
Yes.
Amazing.
The next person to have Jan's surgery will have additional sensors placed in the brain to receive the sensation of touch. The progress is coming rapidly. They're working on a wireless version of the implant to eliminate the connection in the skull. And Dr. Jeffrey Ling told us that the lab experiments will one day enter the real world.
We're going to not stop at just arms and hands. I think that it's going to open the way for things like sight and sound. And my dream that we'll be able to take this into all sorts of patients with stroke, patients with cerebral palsy, and the elderly.
I think when other quadriplegics see what I'm doing with the arm, they're all going to say, oh, wow, I wish I could do that.
That's the way I like to get cookies. Awesome, thank you so much.
I just feel very honored to be the one who gets to do it.
Okay. So one take-home message from this is that when she was describing what it was like to control it, and she said she just thought about it as if she was moving her own arm. I think it's really important in terms of decoding that you have that characteristic. It should be intuitive and easy to use. All right, I just want to show you a few more detailed actions that she can do. We had her do these ARAT tests, which are tests of movement that stroke patients are often assessed with. By the way, this is Jan sitting right here. Here's her wheelchair controller. And you can see how coordinated and smooth and beautiful these movements are, just like real movements. I think you can see it especially in this type of movement. Look at the multi-joint coordination that's going on and how well she can do this. And if you look closely, you can almost see her personality coming through and the way she's gently lays these things down and grasps them. This one, she's picking up a rock, and the thing that's nice about this is that you can actually see all 10 degrees of freedom operating here. And she has to conform to this irregular shape by curving her little finger around the bottom.
Scoops to hold it.
Now, the thing I want to emphasize is that, these weren't just like one-offs where we, repeated this thousands of times and picked the one movie that worked. If you look, these are days, and this is 100 days post-implant, and these are individual testing days, each of these dots. And so we look at success rate here, and you see that there's a classic learning curve taking place over these 100 days. So her success rate on these prescribed tasks increase. If you look at the amount of time it took her to do these ARAT tasks, it continuously decreased. She got faster and faster. And if you look at different metrics of path efficiency, they continuously increased over time. So she does this every day when she comes in the lab, and she gets, at least to a certain point, she got better and better. Okay, one of the other things you can do are spontaneous tasks. So in this case, we're trying to encourage her to use hand shape to learn to shape the hand. So she decided that she would teach me how to play rock, paper, scissors.
What? 2, 3, shoot. Same thing. Aye. Yeah, but my paper was trying to cover his paper. Okay, ready? 1, 2, 3, shoot. Rock.
One of the things that Jan wanted to do was to feed herself chocolate. So one day we decided to let her try that. I'm going to show you 2 days. The first day was the very first time she tried it. And you have to realize that, this thing can generate 50 pounds of force. And so even like when I was standing there, it's risky. And so what we had initially is we didn't let her get anywhere near her face. We had all these safety devices so she wouldn't smash into herself. And so the first time we did this chocolate bar is the first time she ever brought this thing near her face. And she was a little worried about that. And you'll see that the first day, that there's some hesitation in doing that. So this is the very first time we tried it. That's what I'm calling authoritative grip. And you can see she's not so certain that she wants to do this, but she really wants the chocolate. You can actually see her struggling with this. And then she decides to just go for it. There we go. And the next day she has a lot more confidence. She just picks it up and brings it directly to her face.
That worked.
And
There comes drop it on my hand.
again, even while she's chewing, she still has control. She doesn't even realize it.
Drop it.
One small pebble for a woman, one giant bite for BCI.
She's an enthusiastic subject. So just to show you a little bit about what we think about this technology, right now it's a laboratory experiment. It needs a number of technological developments before it's ready for clinical use or even to bring it home. One thing we're going to do in the next subject, coming up in the next month or so, is to give the feedback to the subject. And the way we're going to do this is implant some additional arrays. This time they'll be in sensory cortex, and we'll provide electrical stimulation to the finger regions of sensory cortex that will be driven by tactile receptors on the robotic fingers. We want to, again, use telemetry so we can get rid of these connectors that come through the skin. We want better electrodes. We want better prosthetic devices. And then even we want to eliminate the robots and reanimate the subject's own hand by activating their muscles. And then, of course, there's always room for improvement in our decoding algorithms. So in terms of what we want to do, I think the next big step is to get this into the home. We want to increase the performance. We want to start having more dexterous control. Of course, we're always striving to get to more degrees of freedom. What this will do, if it really works in the home, is alleviate the need for continuous guidance by a caregiver. So, you have to justify this to insurance companies, and we can say, well, instead of paying for 8 hours of assistance, maybe you only need 3 hours, because the subject will be more self-sufficient. And then in terms of a business plan, I think it's informative to think about what happened with deep brain stimulation for Parkinson's disease. Again, that started out as a laboratory university research project, eventually migrated to other universities, and then into the public domain. So now there are 100,000 people walking around with deep brain stimulators in their head. So I think if we can have that kind of pathway for prosthetics, that will be quite successful. So I just want to illustrate who all these people were that helped over the years. This work began at Arizona State University, and then I moved to San Diego Neurosciences Institute, where I met Paul. And then I moved to University of Pittsburgh, and I have a fairly large laboratory where we do the basic research. And then I have a whole set of clinical collaborators, people in physical medicine, rehab, neurosurgeons, bioengineers. And then I also collaborate with a fair number of people at Carnegie Mellon, both at the Robotics Institute and bioengineering there and in the statistics department. Thank you very much.
starting it from.
Thank you. It was really amazing, absolutely, what you can do with the motor cortex signals. Now, do we need a cerebellum then?
Well, you have to realize, again, as I showed in that cartoon, there's billions of neurons that are firing throughout all these tasks, even the prosthetic task. Okay, there's many, and I'm sure there's a large number of those that are active in the cerebellum. They send their output to motor cortex. So part of that motor cortex signal that we're sampling was derived from structures like the cerebellum.
Yeah. I was thinking what you showed about the saccades and wondered whether it could be useful to use gaze tracking or eye tracking as an auxiliary input just to reduce noise. Or,
in fact, that is a device that's often used by people who are paralyzed, especially those who are locked in. But you'll find that if you have any other kind of movement besides eye movement, that the subjects tend to reject that. It's very difficult to use. Oftentimes you look at things that you don't intend to move to or you don't want to because you're constantly saccading around the room. It takes a lot of energy, effort to actually make it work the way they want it to work.
You developed this wonderful arm, and I was just wondering, I didn't understand how then that is implemented in Jan's body, because she's got two arms, and obviously she can't use them. And in order to be able to use the one that you developed, does that mean that one arm is got to be replaced? Or I don't know how this works.
Well, to tell you the truth, we don't have a great understanding of that either. Okay, but it seems even if you take an intact subject like these monkeys who have two great arms that they can use, and we just restrain them and force them to use this, they just relax their arms and can direct their motor cortical signals to operate this device with completely removing the necessity of contracting their muscles. So you have to remember that motor cortex is just one node in this complex network, and that you can easily just facilitate different portions of the network, in this case those that are associated with muscle contraction, but still operate the motor cortex. I think that's what this shows.
Would she be able to use these arms for everyday life?
Yes. Yeah. In fact, what we did in one set of experiments, we actually had her control two arms simultaneously from the same set of neurons. Granted, the ipsilateral arm she didn't have much control over. I mean, it was much worse, but she could still pick up a box with two hands and place it. And so normally, the way it works is motor cortex goes to the opposite arm. So the one that would normally work her left arm, we would conceive of as being the right cortex, and we only had electrodes on her left side.
Thank you very much. I have to admit that I didn't believe in brain-computer, in brain-machine interface. And from now, I'm starting to believe that you can decode the trajectory, 3D trajectory. I have two questions. The first one is about rehabilitation, not restoration. That is like any kind of application for stroke people to retrain the brain, specific area of the brain to do.
There are a number of laboratories that are trying to proceed along that. One thing that's nice about this is that, at least in our hands when we're recording actual action potentials, that these are neurons, okay, in the brain control task, in order for it to actually operate the device, they have to modulate those neurons. And so what you can do, I believe that what people are trying to do is to get them by recording with ECOG or EEG, trying to get them to modulate their neural activity in specific ways that they believe will increase the ability of them to control the residual muscles that they can. So it's a sort of intercepting neural signals that they believe are associated with generating new arm movements and trying to encourage them through biofeedback to do that. That's where the rehab field is going with this, I believe.
And the second question is about the area. How important is to place this electrode in this area? Because it's a very tiny area, and she can do a lot of movement in this tiny area. So I wonder, if you cover more area, what could you be able to do?
Well, we find these very similar signals in all the areas around motor cortex, including sensory cortex, which is right behind the central sulcus. So, in the laboratory, we've recorded from, premotor cortex, parietal cortex. Other people have recorded from cerebellum, and you see very similar signals. Now, we record from motor cortex because we have the most experience with that, and also because the neurons, the cell size in motor cortex are extremely large, and they're easy to record action potentials from. Given that and the fact that motor cortex is easily accessible from the surface, we've gone for that. Now, I think we could probably do fairly well or equally as well by recording from these other areas. And in terms of what these other areas are doing, I think we can capture other things from these other areas. For instance, as we go to manipulation and we want to recognize the type of object that the subject is intending to go to, there may be other areas of the brain like intraparietal sulcus that we can capture better signals related to objects from those areas. So we are interested in going there.
I was a bit puzzled by how you decode the population code that you have. Basically, you have all those vectors, and one of them is proeminent. Could be called a winner vector, let's say.
No, it's not a winner-take-all. We actually consider each neuron to make an equal contribution. So we take all the neurons and we sum them up just algebraically. So this population vector is just a vector sum of the contribution from each neuron. So each neuron, the only weight is how fast the neuron is firing. That is just the length of the vector. So, but each neuron has the capability of contributing throughout its entire range of firing.
I'm over and over again astonished when I see the movies or eating chocolate, and it always seems for me too simple. I mean, you put in some electrodes, you take a linear model, and it could work. So what were the difficulties that you have encountered over the way? What is the key element you had to solve to get this to work?
Well, again, we're lucky, I think, because it seems to be that this cosine tuning property is fairly robust. So it seems to be a principle that we can follow. So it gets us in the ballpark. So we get close, and then the rest of this takes place through training, where she learns to mold her neural activity to fit our model. And that's really the key to success here, is the learning that can take place.
Thank you for your amazing talk. I'd like to know your thought about other means of such as the surface EMG, the eye gaze, EEGs. There's various people working on different modes for acquiring those signals. Do you think your work will be like disruptive like iPhone, where everyone should start working on this to avoid being like Nokia? Or do you think your work will be like Ferrari, where only a few people that really needs this technology will use it, and there will be other modes of acquiring those signals that will be useful for other people?
Well, of course, I'm fairly biased, okay, because what we're doing seems to work so well, and I've spent 30 years working on this. Most people in the field of BCI are doing recordings from either the surface of the scalp, EEGs, or they're doing ECOGs, which you can do with patients who are being studied for epilepsy for a couple of weeks. Those signals, in my view, are far inferior to what we record. There's less information that you can extract from those signals, and very few people can get anything more than basically a binary switch from these sorts of signals. So I believe that to really get high performance, we're going to need to do this kind of intracranial extracellular recording of the microelectrodes. Now, again, how do we see this proceeding? If you ask a neurosurgeon how difficult the surgery is on a scale of 1 to 10, most of them would say maybe a 1 or 2. As we increase the technology to make these electrodes easier to implant by giving them flexible ribbon cables and telemetry, this could become very routine. So the entire procedure could take maybe an hour and a half surgery, which is considered relatively minor. If you look at how long it takes to place a deep brain stimulator, that's a 3 to 8 hour operation. And there you're putting a much larger electrode deep into the brain. These things are basically like little pieces of Velcro that you put on top of the brain. So in terms of severity of surgery, it's actually pretty minor. And we think that as this gets spread throughout the academic community and more surgeons are trained in this technique, that it'll become more commonplace and well accepted.
I'm curious about the next step, the tactile and possibly the proprioceptive sensory input. So if I understood correctly, it took like decades to decode the brain signals and then translate this into a robot movement. What needs to be done and learned about the sensory input for the brain to make sense of what you feed into the sensory cortex?
Well, we're at the very beginning of that right now. So what we're going to do is just put in microstimulation in the area of brain that's related to finger sensation. We don't really, electrical stimulation is highly artificial, so we don't really know what the percept of that's going to be. And when you do this in monkeys, you can't ask the monkeys what they felt. So, I view this as a very crude first step, but at least we'll be able to ask the subjects what they're feeling when we do this. So, we can vary different parameters, amount of current, frequency of current, pattern of current, and begin to get an idea of how this might work. But we're at the very beginning stages, and I don't want to, say that we're going to even impart natural sensation. It could be some feeling of buzzing that occurs, but that could still be informative for the subject. So we're at the very beginning stages, and I don't make any big claims about this.
So looking forward, do you see this as a new way of doing teleoperation? Or, is that something that, for instance, DARPA are interested in?
Well, there are people out there that talk about that. But, right now, physical movements that you can actually make are much better than what we're able to decode. And we need to go well beyond what we've done so far to get a more elaborate signal that will even approximate what we can really do with our hands and arms. As far as, a lot of people talk about science fiction of enhancement, like memory storage and better cognitive powers. The point is we need to do the science first so we understand what these things mean and how they're represented in the brain. We have no idea at this point. So I keep, these futuristic ideas, I think, still remain in the realm of science fiction, as far as I'm concerned.
May I ask you a practical question in terms of the electrode arrays? For how long can you keep them in the cortex without the need of replacing them because of biofouling or something?
So right now with the Utah electrode arrays, I would say that we get really great signals for about 6 to 9 months after implant. And after that, you see a gradual deterioration in the recording quality. Oftentimes, you can leave these arrays in for years and still pick up signals. They're just not particularly high quality. And that's what we've seen with Jan so far. So now she's, originally she's only supposed to have this in for 1 year, but she elected to go beyond that 1 year. And so we're in our actually completing our second year now. And the signals have deteriorated so that now instead of doing 10 degree of freedom movements, we're doing 5 degree of freedom movements. And that's probably about as good as we can do with the signals we have. But there's technology on the near horizon that's going to try to address these issues of, the deterioration we see around the electrode. So we feel confident that in the near future, these electrode longevity will increase dramatically.
So Andy, just to finish up, a very practical question. So in some sense, you're measuring from a brain with intrinsic encoding capabilities and an ability to control action of the body as it is, right? But now you put it in this closed loop system where it can get feedback, so you start to also capitalize on abilities of learning. So actually, you look at these two systems now interacting. So what do you think is the most important component that you're tapping into? It is learning capability of the brain to, in some sense, perform any mapping that you expose it to, or are you tapping into these intrinsic abilities to control action?
Well, I think it's important. Of course, you need both. But in order to let the learning happen, you have to be close to start. You have to approximate, because the subject has to have some metric of success. If learning requires, you try a variety of things and you see what works. And if there's no success, okay, then they're not going to learn. Right. And I think the closer, the easier you can make it for them to learn, the farther you can go.
Okay, excellent. Well, Andy, thanks again for a fantastic talk. And Holger will be chairing our next session. Oh, and
Yeah. Okay. Thanks, Paul. Thanks, Andrew, again. That was really amazing. I suppose that one of the major aspects here is that the system was working under closed-loop conditions, and it helps a lot training and learning. Now, I think in the next session,